Interestingly, we have seen that dual CAR T, but this is bicistronic CAR T. It means that they engineer T cells with two different CARs simultaneously expressing two different CARs, but with using single vector that targets two antigens. So, because as most of you are aware that FGFR and B7H3 is highly expressed in most of the childhood cancers, especially in rhabdomyosarcoma. So, just a minute. So, yeah, so we'll begin. Oh, sorry. So, as we all know that rhabdomyosarcoma is covering around 3 to 4% of the childhood cancer, and challenges in CAR Ts with solid tumors are because of antigen heterogeneity, T cell exhaustion, and poor persistence. So all these points, all these three challenges are covered in this paper, and how to improve and how to overcome these challenges they have mentioned. So what they did, they have used two different antigens, FGFR4, which is extensively expressed in rhabdomyosarcoma, and CD276, which is B7H3. So they have used these two antigens to overcome tumor antigenicity and also overcome tumor escape. So that FGFR4 expression, especially in fusion positive rhabdomyosarcoma, is seen, and around 10% of fusion negative rhabdomyosarcoma also activates FGFR4. So, along with that, there is very low expression in normal tissue, so it makes it a viable target. Then CD276 or B7H3 is also overexpressed in RMS, along with other solid tumors also, and it is upregulated by that PAX3 FOXO1 fusion. So, because of which they use these two these two antigens. So, what they modify here is because most of the second generation CAR use CD8 as hinge transmembrane and 41BB. Okay, so this is what most of the second generation CAR T. Is my pointer visible?
Yes, visible.
Yeah. So what they did is with FGFR4, they have used CD28 as hinge transmembrane protein, along with that CD28 as co-stimulatory domain. And with B7H3, they used 41BB along with CD8 domain. So what they noticed here is there is something called leg volume and total flux. So what they noticed is when they use these two uh, bicistronic CAR T, a significant reduction in leg volume means significant reduction in tumor volume, and say along with that tumor flux. Along with that, they have noticed that the inflammatory cytokines production is very high, uh, which helps in eradication of the tumor. So also they have used two types of xenograft model. One is RH30 and other is RH559. So RH30 model was fusion positive RMS. RMS 559 was fusion negative RMS. So because it was already discovered that this FGFR4 with that conventional CAR T, it delays tumor growth, but it does not eradicate the tumor. So because of which they modified this, and B7H3 CAR T they used uh, to increase the efficacy of CAR T because it increases TCR signaling and synergistic T cell activation. So finally, they noted that with the using of these two by antigens in bisystronic CAR T, they found that almost all the tumor is eradicated with uh, there was no challenge with tumor heterogeneity. Persistence of CAR T was significantly better when they used single tumor specific CAR T or CAR T, dual CAR T with conventional method like CD8 and 41BBS domain. So this is what is very interesting. That's why I bring this paper to everyone's notice. So, uh, Dr. Prerna, any comments on this?